Hello, and welcome to today's APS webinar entitled Leveraging Novel Animal Models for Translational Research, presented in partnership with Fauna Bio. My name is Jacob White of the American Physiological Society, and I'll be serving as your moderator today. Now, in today's session, Dr. Lara de Almaro Silva will present her research on understanding physiological mechanisms that allow animals in nature to overcome stressors that cause major metabolic challenges in the brain. And Dr. Diana Sarko will present advantages and experimental design considerations of using naked mole rats as a research model for longevity, cancer resistance, and sensory specializations. Now, a few housekeeping notes. Out of respect for our speakers, we ask that you refrain from photo or video captures of the presentations in whole or in part. This session will be recorded and available for future viewing through a link on the registration webpage. Now, recording or taking photographs of another presenter's information without their explicit permission is prohibited. At this time, all attendees, except for our speakers, are in listen-only mode with video off. We are happy to answer any questions that you may have after the presentations at the end of the session. Questions may be submitted using the Q&A function at the bottom of your screen. The session will end at approximately 12 p.m. Eastern Time. Now, before we begin, I'd like to thank today's event sponsor, Fauna Bio, for their support of our Comparative and Evolutionary Physiology Section Awards and Trainee Breakfast. Now I'd like to invite Dr. Ben Sajak, Director of Emerging Animal Models at Fauna Bio, to present a brief update on the exciting work that they're doing. Ben, whenever you're ready.
All right, thank you. Yeah, thank you, Jacob. Thank you, everyone, for attending today. I just wanted to give a brief introduction of what we do at Fauna Bio as it relates to today's topic. Indeed, this is what we do, leverage novel animal models for translational research at Fauna Bio. So we leverage millions of years of evolution in that it's developed species that have profound disease resistance against conditions that ail us humans. So we can study these disease-resistant strategies and evaluate the shared genes we have with these species to leverage into new understanding to improve human health. So we do this by using our discovery platform called Convergence, which unlocks novel treatment mechanisms from extreme animal physiology as well as adaptations by integrating human data at every step. So the very high-level overview of this process is we find and identify protective signatures across extreme animal adaptations and data sets, compare those to human data. We map those results to human genomics and validate the targets in human cell models or in rodents in vivo. These rodents wouldn't have those adaptations. And then finally predict compounds that mimic that natural protection, or in some cases create our own compounds, and usually test those in human cell lines first. And then ideally move it on to the clinic for drug development. So my job is primarily focused on this first component here, identifying unique species with protective signatures and getting those extreme animal adaptation data sets. So I wanted to highlight a couple species that we use at Fauna Bio. The bulk of our data is from the hibernating 13-line ground squirrel, which has a remarkable resistance and reversal of many human-like disease pathologies because it's a natural and obligate hibernator. 6 months out of the year it hibernates, and it's a system-wide shutdown of sorts for 2 weeks at a time. It'll reduce its heart rate from 300 beats per minute to 3 beats per minute. It barely takes any breath, and through this lack of blood flow and oxygen throughout organ systems, it is able to sort of not only survive but thrive in these conditions and there's a lot of disease avoidance relevance happening throughout these systems here. And so I've highlighted a few areas with active projects that we're working on. In neurodegeneration, it's sort of turned into a sort of a retinal disease project, as well as studying fibrosis of the heart and recovery from fibrosis, as well as pulmonary fibrosis. And then we have a partnership with Eli Lilly, looking into obesity phenotypes because they gorge themselves in preparation for hibernation and subsequently lose that throughout the hibernation cycle and avoid consequences related to their induced obesity. So it's a nice natural model for that. But I want to take the rest of my time to talk about a newer project we have going on with spiny mice in collaboration with the University of Florida. Spiny mice, you may know, have remarkable adaptations in tissue regeneration. So they have this common theme of reduced scarring and reduced inflammatory response, with several organ damage models being now characterized. Most commonly studied is skin regeneration, which is shown on the left here. You can see if you take a pretty dramatic dermal punch, that grows back within just a couple weeks without scarring, and hair follicles and everything form as if that damage never happened. And it turns out if you apply a similar damage model to organ systems, you get a similar phenomenon with reduced inflammation, reduced damage, and better functional recovery, whether it's the kidney, CNS, or the heart. So we've been really interested in the new CNS data coming out with this species compared to lab mouse here. So there's this really interesting paper that came out a couple of years ago. They damaged the spinal cord and showed that there's better axonal recovery within that damaged site, and thereby better functional recovery from an otherwise pretty traumatic spinal cord injury to a lab mouse. And I encourage you to check out that paper. They have a really striking video where they show the comparison between the lab mouse not being able to walk with its hind limbs and the spiny mouse being able to do so after the same injury. And so down below is a schematic of what we think is going on here. Reduced reactive astrocyte activity and microglia activity, leading to better neuronal recovery from that damage. So with Dr. Janak Gair from the University of Florida, we're identifying targets that are at the root cause of this reduced CNS inflammation. So with this model, we're using a foreign body response damage model here. So injecting a neuronal probe into the cortex of CD1 mice and spiny mice, and then collecting those tissues at several time points after that damage response, sequencing with RNA-seq, bulk and sometimes single cell, and then looking at some of the changes. So we've seen what we've expected to see, which is decreased inflammation sort of themes throughout the data sets. We're seeing surprisingly increased astrocyte marker activity in the spiny mice compared to lab mice in some of these early time points, as well as the late time point, which is pretty interesting here. And then we can dig a little deeper and look at specific potential targets that are that may be involved in a protective mechanism of sorts with the spiny mouse, or an sort of over inflammatory response in the lab mouse by comparison. So we're just digging into this data, but it's been an exciting and fruitful collaboration so far. So those are just two really brief examples, but there's vast potential for this type of work through collaboration. I've highlighted here about 400 species with whole genome sequencing. And if you just do the hard work and look at the literature through these species, we've been able to find well over 50 disease-resistant species to this point in our own sort of investigation. And these are sometimes one-off papers that show some strong potential in a disease avoidance strategy or tissue recovery, something like that. So there's just a lot of potential here. And so what we do is we collaborate with these institutions that have experts and labs in these species um, to do novel treatment discovery through our platform. So that's why we like supporting APS. It connects us to this great research going on in this field. We could go to disease-specific conferences, but we really like APS because they really support this mission of non-traditional and novel animal model use for human health. It's often difficult to study these types of species, as we all know, and it really requires not only institutional support, but grassroots communities support from each other and tool sharing. And these are all efforts that we hope to continue to support through APS and other organizations. So as Jacob mentioned, we support a translational research award as well as a trainee breakfast, and we hope to continue supporting webinars like this one in the future. So hopefully we'll see you in Baltimore. Thank you very much.
Thank you so much, Dr. Sajak. Now, before we begin, I just have a quick poll that I want to launch for the audience regarding interest in collaboration with Fauna Bio. So that should pop up on your screens. And please feel free just to take a moment to answer a couple of questions. And then we'll get started with our speaker introductions. A few more seconds and then we'll close. Well, thank you for everyone who participated. Now it is my pleasure to introduce our speakers for today. Our first speaker, it will be Lara del Amaral Silva, who is an assistant professor of neurobiology at Wake Forest University. She investigates neuroprotective mechanisms that birds evolved against hypoxia and insulin resistance, and also dedicates much of her time to mentoring the next generation of scientists. And Diana Sarko, who is an associate professor at Southern Illinois University School of Medicine. Her research interests includes somatosensory and audiovisual processing. Now, on to our program. Lara, please begin whenever you're ready.
Jacob, can you see my screen?
Yep, you're good to go.
Awesome. So thank you, Jacob, for the introduction. I would like also to thank APS and Fauna Bio for the invitation for this talk. So I'm an early career professor starting my own lab journey using some of my basic comparative research interest and experience to start my own lab. Today, I'll talk a little bit about some of basic comparative research translated all the way to the clinics and drugs that we use daily in the clinic. Then I'll talk about the importance of comparative models for neuroscience, and I'll finish with some of the projects we are developing in my lab since last year. Okay, so there you go. So this is a traditional flow chart of drug discovery. Sorry, I'm having trouble to see my own slides. Of drug discovery. So usually the disease is identified on the clinic, and then researchers develop usually a rodent model for that disease. So further targets of the disease can be identified. And some procedures and compounds can be tested to ameliorate that disease. And if these compounds work, then this will go forward for preclinical studies to understand interactions and the safety of these compounds. And then if it works, it goes to clinical trials on phase 1, 2, and 3, which can fail all the way in one of these phases. And if that works in the clinics, this drug will be reviewed and approved for be widely sold for the treatment of the disease. However, doing things in only one way keep researchers away from possibilities beyond this scope and finding solutions for problems that are still not resolved. So another way to look at these problems is to look back on evolution and see how some animals already overcome conditions that cause disease in humans, following the August Crow principle, that for a large number of problems, there will be some animal of choice or a few of such animals in which it can be mostly conveniently studied. So for today, I wanted to bring a very classical example that is used on clinics for many years now, so we can all identify with some of these drugs, and then I'll have a contemporary example for us. So the first example I brought is started on the curiosity-driven research on the toxicology of snake bites, so specifically the Brazilian lance head snake. And studying the components of this toxicology, this led to the research of what these compounds did. So Beraldo and Roosevelt, they were able to isolate bradykinin as the compound of the snake bite that cause vasodilation in their prey. And then further on this research, Ferreira in the 60s discovered a potentiating peptide for the bradykinin that keeps this vasodilation for a long time on the prey. And going forward with this research, two groups were studying very different things. So the first group was trying to understand the angiotensin conversion in pulmonary circulation at a basic level. And Ferreira went forward with these compound studies on bradykinin and saw that these bradykinin disappeared in the pulmonary circulation. So later on, these groups worked together and they saw that bradykinin actually inhibit angiotensin conversion. Let me put my laser here. Inhibit angiotensin conversion on the pulmonary circulation, and that's why they saw this big vasodilation in the prey. And this was the foundation for Squibb Biotech to isolate and optimize the compound and create the ACE inhibitors that are widely prescribed on the clinic for high blood pressure, heart failure, and heart failure prevention. And I wanted to bring another very contemporary example of curiosity-driven research that went all the way to the clinic. So this research started with the Gila monster saliva. So first, Rothman and Pisano saw that the Gila monster saliva spike amylase in guinea pig pancreas, and then they worked to isolate the compound that would make that. So then they were able to isolate exendin-4, which was a GLP-1-like amino acid that was an agonist for the GLP-1 receptors, but remained in the bloodstream for more than 2 hours, so had like a prolonged effect on the Gila monster prey. And right away, they saw the potential of this compound to increase insulin and deal with high glucose problems in diabetes. So they pitched this compound for many big pharmaceuticals, but they didn't want to be involved with a lizard compound. So in the 90s, startup, Amylin Pharmaceuticals, took this compound in and they developed a synthetic version of it, which indeed increased insulin on the plasma and normalized the glucose levels in mice. This compound was then transformed in the exenatide drug, which was FDA approved for diabetes and had a side effect of a small weight loss. So then Novo Nordisk, trying to compete with this diabetes treatment, tried to make their own synthetic compound, GLP-1 agonist, and they optimized that to avoid rapid degradation. So the patient would have like less injections per day. And they did develop liraglutide, which was approved to treat diabetes. And they saw the side effect of weight loss. So then they investigated the side effect of this diabetes treatment, and they saw that these GLP-1 agonists also bind to hypothalamic neurons. So they optimized these agonists to bind more to hypothalamic neurons, and this was improved on the semaglutide that was approved in 2001, and since then is widely prescribed for diabetes and for weight loss. And then we have the advancement of this research that target not only GLP-1 receptors, but GLP receptors, and now even glucagon receptors, which seems to be more effective and is in clinical trials. So I wanted to bring two very relatable examples of these potential of comparative research models, but there are a lot of examples, just like Ben was talking to us about the regenerative potential of spiny mouse and researches on hybridization with potential for avoiding muscle degradation, and many other examples. Of course, I'm not reading all of them. And I think it's a very nice, we are in a very nice timeline that some companies, much like FaunaBio, start to look at these weird animals to find targets to overcome diseases. And not only FaunaBio, but of course, some of their competitors like Pell Therapeutics are looking for that. So I think a lot of advancement will come in the next years. And I'm really happy about it. But what about neuroscience? So neuroscience is intrinsically related with comparative models. So the first action potential was able to be recorded intracellularly due to this model of a giant axon in a squid. So they were able to put an electrode in and record the action potential. And later, this same model allowed Kenneth Cole to develop the voltage clamp. And we now know the currents that are necessary to elicit an action potential. And this gave the Nobel Prize to Hodgkin Huxley in 1963. Another Nobel Prize winning comparative model in neuroscience is the aplysia. So the sea slug have this reduced nervous system with around like 100 neurons, and because they have this reduced nervous system, and they were able to have complex behaviors like associative and non-associative memory. So going back to the model of the dog salivating before food, we were able to understand on how these mechanisms work together to create a memory. And this gave Eric Kandel the Nobel Prize on the 2000s. And beyond these very classic models of comparative neurophysiology and neuroscience, there are many others that allow us to understand how things work. So we understand better how circuit dynamics because the somatogastric ganglia studies in crabs and lobsters. So all the rhythm generator models and how they can interact to form a circuit, degeneracy, among many others. The most popular neurophysiological model for vocal learning and memory nowadays are songbirds, which can even infer in speech pathologies. Spatial navigation is better understood because of bat models, and face recognition is studied in paper wasps, which can also recognize face of their mates and have a reduced nervous system. So there are many other examples like that on comparative physiology inferring on neuroscience, so much so that the Advisory Council for NINDS at NIH recognized that the overemphasis of rodent models is an obstacle to the advancement of new areas of fundamental neuroscience, and they encourage prioritizing basic biology and neuronal metabolism for the development of fundamental neuroscience, and they encourage exploratory research and the use of non-traditional organisms for such. Awesome. So all of that, what about my lab. So as a title in the background, the first slide would hint, I consider animals that overcome metabolic challenge to the brain to search for mechanisms to overcome brain diseases. So a brief recap here, I think we are all familiar with the concept that the brain is this complex, highly active, and plastic organ that controls most behaviors. And for that, it uses an outstanding amount of energy. So in a human, it occupies around 2% of the body size, while consuming 20% of the energy and 25% of the glucose that we of our intake. And for that, it relies on the mitochondrial respiration, so the ATP here coming from the mitochondrial respiration, and of course, oxygen as the final acceptors of electron in the electron transport chain. So as a rule, when there is no oxygen, there is no function. And how function stops? So first I will show an example, a very brief example, simplified, on how a neuron that have all its energetic needs met would function. And then I'll talk about this loss of function. So here, when the neuron is supplied with all its energetic needs, an action potential arrive here to the presynaptic terminal. The polarization of the presynaptic terminal will allow to some synaptic vesicles to fuse here to the synaptic cleft, releasing glutamate. This glutamate will bind here to glutamatergic postsynaptic receptors, so AMPA and NMDA, cause an influx of sodium and calcium. These positive ions would depolarize this postsynaptic neuron, which would fire action potentials. And after that, this glutamate here in the synaptic cleft is rapidly removed and largely recycled between astrocytes and the presynaptic neuron. And this cycle would go over again and again and again. However, when there is no oxygen, the function stops through a neurotoxic cascade. So this starts with a reverse operation of glutamate transporters, so they would not uptake glutamate anymore. Instead, they would release more glutamate here in the synaptic cleft. This would cause an overstimulation of these AMPA and NMDA receptors that would cause a big inflow of sodium and calcium. With this big inflow of positive ions, the cell depolarize and fire a lot of action potentials and depletes ATP because anaerobic glycolysis is not enough to produce ATP for this neuron anymore. And with ATP depletion, the sodium-potassium pump stops, so the ion concentration cannot be restored, and this neuron gets to the polarization to a non-returning point. At the same time, the mitochondria here on the postsynaptic neuron receive all this calcium coming from NMDA receptors, which trigger apoptotic factors and kills these neurons and all neurons around it. So unfortunately, this is a very common issue arising in multiple disorders, so that being by stroke, a heart attack that doesn't pump blood to the brain, brain traumatic injury, infections like COVID that provide silent hypoxia, overdose. The list goes on and on and on, with a common denominator of this big energetic stress. And those are leading cause of death and disabilities worldwide, and even the sublethal energetic stress that can come from an improper energetic supply of these neurons are related to the onset and progression of conditions like Alzheimer, Parkinson, Huntington's, ALS, and even neuropsychiatric disorders. So there is a lot of conditions that have this root on this energetic stress. And in parallel to that, there is this big translational block of neuroprotective mechanisms of disease models to be translated to the clinic. So there is a necessity to think outside of the box and look for alternative models to find solutions for these big problems. So this is where my lab focused. So we believe we can learn from animals that overcome metabolic challenge to the brain to gain overarching insights into brain diseases. And I want to acknowledge that this is an area that have some comparative models studied. usually animals that need to face hypoxia on their environment, they do have some adaptations to overcome that situations that they face daily, being that because they are swimming in ponds that have very little oxygen, because they hibernate in these ponds with very little oxygen, because they dive deep so they can't breathe all the time, again, hibernation, or they live in burrows like the naked mole rat, which we'll learn more about later, that have low availability of oxygen. Including here the bullfrog model that I studied during my postdoc. We can transform the neural circuits to be resistant to this hypoxia after hibernation. And I studied this during my postdoc, and we wrote a review for the physiology, which was the physiology cover of November in the American Physiological Society journal. So I brought it here in case you guys have any interest about that. But for my own lab, I had studied birds for around 10 years before my postdoc, where I could study this frog model. So I wanted to go back for my root questions to understand how birds, the most energetically expensive vertebrates with the highest body temperature and the highest metabolic rate, can present superior tolerance to hypoxia compared to mammals. And this doesn't seem to be only because of peripheral adjustments. So they have a better lung function, better oxygen extraction in the lungs, or adjustment in the cardiovascular system. But this seemed to be also true for the brain. So this is a recording of cerebellar activity. There is not much research on the topic, but we can see these recordings showing that cerebellar activity can be maintained in ducks when there is no oxygen provided to this tissue. And if mice brains are submitted to the same conditions, they will not function at all. So ducks and chickens can have two models. So they maintain cerebellar activity, or they can shut it down and recover activity later on. And this happens to around 70% of the samples. So I hypothesized in my own lab that these animals, even if they didn't go through these big challenges, just big hypoxia challenges throughout life, like these comparative models that I showed before, they would have neuroprotective mechanisms to sustain this highly energetically requiring function, even though they had no oxygen supply, which of course in humans are the root of many neurological diseases. And this is indeed what we are seeing in the lab. So this is a recording, and we have been seeing this consistently in our cells. So this neuron, I patch a neuron, so it's patch clamp, and we can see the electrophysiology of it. So I inject a little bit of current, and these neuron fire action potentials, and the neuron fired tonically here. And when we put 0% of oxygen in the medium, instead of having this anoxic depolarization showing mammals, the neuron actually hyperpolarized and was kept with this membrane potential for as long as we can record this neuron. So here we lost the cell because it's a really long recording for patch clamp. And here, these bleeps that we are seeing, I was actually injecting current to see that even though this cell is silent, if it could fire action potentials. And we saw that even though the cell is silent, when we inject current, the cell can still fire single action potentials. I did have some cells that did recover from that and start firing tonically after that, but I'm starting my own lab, and we are working on that. So for that question, my first PhD student, Tokai Zhang, is patching intermediate midwell mesopallium cells in chicken chicks brain. And we are investigating the first steps of this neurotoxic cascade that could be avoided to avoid these anoxic depolarization and the neurotoxicity. So, because this doesn't seem to happen here. So we hypothesize that this neuron could have a glutamate transporter upregulation, or the glutamate transporter can be maintained in these hypoxic conditions. Alternatively, there could be an excitation shutdown to decrease the amount of positive ions that come into the postsynaptic neuron, or even inhibitory modulation of this postsynaptic signaling by an inflow of negative ions. So Tokai is working on that. And we do recognize that synapses are the most expensive process in the brain. So we want to understand if this very expensive synaptic signaling could be lost in hypoxia. And we also have plans to try long-term potentiation and understand if this signaling would be lost. And for that, Mehrad, my brand new PhD student that just arrived last week for the spring, will stimulate neurons here, presynaptic neurons, and record from postsynaptic neurons to see the synaptic currents and understand if this could be lost in hypoxia. At the same time, we want to see if these neuroprotective mechanisms would work in vivo. So we are exposing chicken cheeks in vivo to hypoxia. And Alice, one of my undergrad students, is looking for neuronal survivability. So these are her histological slices. Ione, another undergrad student, is working on a behavioral model for learning and memory. So this is an imprinting model, and we want to understand if this capability could be kept after hypoxic exposure. And Alisa, another undergrad student, is looking for hypoxia-inducible genes or genes that could be lost regarding learning and memory in these cheeks. So I wish I had more preliminary data to share with you, but again, I'm starting my own lab and I'm very proud to show the timeline that we had this academic year. During the summer, my lab was still being renewed, and now we have a fully functional lab and we have the students trained, and we are so ready to produce much more quality data. So with that, I would like to thank my team, so my graduate students, undergrads, and even work student. I'd like to thank American Physiological Society and Fauna Bio for the invitation for this talk, and Wake Forest University for a pilot funding that I just got. So thank you.
Thank you so much, Dr. Amaro Silva. And now we will hear from Dr. Diana Sarko. Diana, whenever you're ready.
Thank you. Is that looking okay, Jacob?
Yep, looks great.
Thank you. All right. I wanted to thank Jacob for that wonderful introduction and Laura for that wonderful talk that I get to follow, and the American Physiological Society for the invitation for speaking today along with Fauna Bio. This is a wonderful event, and I love talking about unusual animal models. So my talk today is going to focus on our animal model, the naked mole rat. So just a brief background about some of the sort of anatomical and physiological and neuroanatomical adaptations related to naked mole rats. These are rodents. They are mouse-sized rodents, and they are unusual in a number of ways. If you look at their habitat, they live mostly in Eastern Africa. They are subterranean, so they live in these underground burrows that can be very extensive, so up to about 3 kilometers long in the wild. They are also, within these subterranean environments, very tolerant of these hypoxic and hypercapnic conditions, which has been shown previously by Dr. Thomas Park et al. In 2017. So very adapted to those conditions. Also poor regulators of body temperature. They're essentially considered to be cold-blooded, so they mostly sort of take on the ambient temperature of their surroundings. That's something to also keep in mind in terms of housing these unusual animals. They really need very warm and very humid environmental conditions for their housing. In addition to those aspects, they have this incredible longevity. They live to be about 40 years old, possibly longer, based on some animals in these longer term colonies of Shelly Buffensteins. And so this is over 10 times as long as you would expect for, say, a mouse or a rat. So this incredible longevity, they're used in aging research, also in cancer studies, because they live that long, mostly without getting cancer as well. Along with that longevity, they have a long gestation period and weaning period. And they also have this unique social structure that makes them an interesting animal model as well. So they are eusocial like bees, and they have a queen along with usually 1 to 3 male breeders typically, and then non-breeders also within the colony. And that average colony size is typically around 80 animals, but can be up to about 300 in the wild as well. So this really complex social hierarchy and social structure for these animals. We are especially interested in these animals for our lab in terms of some of their sensory and motor adaptations. So these animals are a bit unusual also in that they have these very prominent continuously erupting incisors that are flexible for these lower incisors. So you can see these animals have a very wide gape, and they also have a lot of jaw-related musculature. So a previous study back in 1992 estimated that the head musculature is about 25% of the total musculature of this animal. And you can see how large these muscles of mastication are in the naked mole rat in this postmortem dissection. So this very large and very medially extensive temporalis muscle, along with this very large masseter muscle. And you can compare that to a mouse, where you can see the smaller temporalis, smaller masseter muscle, and how more laterally located that temporalis is. It's not extending all the way up to that midline in these postmortem dissections. So very impressive head musculature. That 25% of the total muscle mass, that's equivalent to our leg musculature for humans. So it's a huge amount of musculature just in that head to kind of help them with digging and excavating underground and exploring and navigating underground. But again, in addition to that, they have these lower incisors that are independently movable. So the upper incisors stay in place, but those lower incisors are able to move independently. So these pictures are a little bit hard to get your bearings on, but we have a probe here, and that is covered with some baby food for the naked mole rats to investigate. These are the lower incisors, so we're looking at the animal kind of head on. You can see the nose here, the upper incisors, and those two lower incisors along with the food probe. So they're able to separate those lower incisors laterally, and then also a bit of a front and back motion, so they can move the teeth rostrally and laterally. They're able to do that through flexible mandibular symphysis. So in humans, that is pretty well fused, of course, between left and right hemimandibles, but for naked mole rats, that's a flexible mandibular symphysis. So the musculature of the neck is what we thought would be sort of helping to control the movements of that. And through some studies by one of our fantastic undergraduate students, Lisa Eckel, she presented this just at the last Society for Neuroscience conference back in October. She did some deeply anesthetized preparations where she would stimulate the musculature of the neck. And she found that a couple of these muscles are helping with those lateral movements. So the anterior digastric and also this transverse mandibular muscle. So we still have, those are very preliminary investigations so far. We still have some additional avenues that we're exploring with that. We want to test additional groups of muscles, especially in combination, to try to find which musculature might be controlling the rostrocaudal movements, as we haven't been able to narrow that down quite yet. In addition to that, we want to do some behavioral experiments, particularly with something like deep lab cut, and be able to characterize kind of the full range of motion of what these naked mole rats are capable of in terms of moving those lower incisors independently. And in addition to that, we're interested in doing some intracortical microstimulation of the motor cortex, which has not yet been mapped out in naked mole rats, to see what we would expect to be a large representation for these lower incisors as they're using those to be able to investigate their environment. So as part of that, in a previous manuscript, I used this sort of chart that's based on what's been shown for whisking in animals that can whisk, so actively move their whiskers side to side to be able to investigate their environment. So things like that, and what we assume might apply to the lower incisors, would be some extra proprioceptive input to be able to sense that environment, need to know where those teeth are in space, and be able to have that extra sensory information come in and then be routed through motor cortex in order to provide sort of better and more accurate motor outputs. So this is a network that we posited based on some cerebellar studies looking at representation of the incisors using electrophysiology in some previous studies. So with that impressive head musculature, one of the things we became interested in and wrote a paper about back in 2019 was just how strong these naked mole rats can bite. So what is their bite force? And we wanted to also look at that within a bit of a social context, so looking at dominant versus subordinate animals, and then also looking through the literature to see how naked mole rats sort of stacked up to other species in which bite force has been ascertained. So we looked at that bite latency and found that subordinate naked mole rats had a shorter bite latency than the dominant animals. That wasn't quite what we expected. We thought the dominant animals would be the ones that are quicker to bite, but we feel like the dominant animals sort of have their place within the social hierarchy and don't feel like they have as much to prove, maybe. this is what we found for the average bite force. And what we found that was probably most impressive was as we looked across bite force across all these different species, if you took the naked mole rat bite force and compared that to for what you would expect for their body size, then what we found was that their bite force was equivalent to Tasmanian devils. So very, very strong bite force, which is what you might expect given all of that massive head musculature and that wide gape to see what they're capable of. In addition to that, again, with that sensory innervation to the lower incisors, that's never been fully characterized. And we do believe that the lower incisors, because they can move independently, they might be endowed with sort of a greater level and degree of innervation to be able to take in all of that extra somatosensory and tactile input and be able to convert that back to better and more accurate behaviors and investigation of the environment. So again, with some studies done by a couple of our wonderful undergrads in particular, so Lisa Eccles and Savannah Staley helped with these studies, especially Savannah looking through, and we first did a gross dissection to look at some of the innervation that goes to the upper incisor in part and to the lower incisor. So that superior alveolar nerve, which in part gives innervation to the upper incisor, and then that inferior alveolar nerve, which in part gives innervation to the lower incisor. And so just with preliminary data and kind of gross characterization of this, what we found was that for the superior alveolar nerve, it was about 1,000 axons when we did this electron microscopy preparation and counted those axons. And for the inferior alveolar nerve, again, in part going to the lower incisor, it was over 3 times that amount. And this amount of about 3,500 axons is also impressively equivalent to what's been shown in rats. So in a study by Naftel et al. In 1999, they quantified the axonal innervation to the incisors by decalcifying the mandible, cutting through, which is what we've started to do here as well for the naked mole rat, and be able to find these little fiber bundles that are more specific to that lower incisor in particular. And when they quantified that, when they looked at the inferior alveolar nerve, they found that that innervation was about 3,500 axons. So naked mole rats are about 1/7 the body size of rats. So for them to have about an equal amount of innervation in that inferior alveolar nerve is very impressive, since again, rats are about 7 times as big. So to us, this indicates a high degree of lower incisor innervation for their body size and greater orofacial sensory motor control. In terms of their other anatomical and sensory features, again, our lab is kind of especially interested in tactile or somatosensory processing, but I'm also very interested in how the tactile sensory modality doesn't work alone. So as we perceive our environment, we're mostly not perceiving one sort of sensory modality at a time. We have to take in multiple sensory modalities and make sense of that and be able to navigate our environment. So these are the sort of principal sensory modalities and what they're good at. So tactile somatosensory is a big sensory modality for these guys, even though they are considered naked in that they don't have fur, so pelage fur to keep them warm. They do have these tactile hairs, which are well innervated guard hairs that are all over their body. And that helps them to be able to sense their underground subterranean environment and sense the things surrounding it, especially because they don't have very good use of visual cues. So naked mole rats are considered functionally blind. They have these reduced visual systems, but they can actually detect and respond to changes in luminance. So if you go into the naked mole rat room where we have them in the vivarium and you turn the lights on, all of a sudden they will sort of startle and react. So they certainly can tell changes in light intensity. They just probably can't tell one naked mole rat's face from another or one caretaker's face from another. But again, these tactile hairs all over their body really help them to sense their environment. And in previous studies back in 2003, it was shown that with these about 80 tactile hairs distributed over the entire body, that even if you deflect a single hair, they orient to that stimulus very accurately. So very sensitive sensory array on their body, in addition to these prominent incisors that are also very sensitive and take up a huge amount of somatosensory cortex, which I'll talk about next. In addition to that, their auditory sense is somewhat similar to visual in that it's not as well developed. So they do not have an external ear, a pinna, but they do have this little raised annulus that you can see here and tiny over here. And you can also see just how tiny their eyes are there as well. So in terms of their auditory processing, they're not very good at auditory localization of signals. They are attuned to low frequencies, which is what you might expect given their underground subterranean environment. Higher frequencies would get clipped in those underground tunnels. So they're especially attuned to about 4 kilohertz. But they do have a distinct kind of vocal repertoire, and they have distinct colony-specific dialects that have more recently been shown by Barker et al. In 2021. So these are colony-specific, kind of culturally transmitted. So they do have, with that complex eusocial kind of social system and structure and hierarchy, they do also have ways to identify one colony from another. And that seems to be well-developed kind of communication as well. So in terms of that somatosensory or tactile system, some previous studies by Ken Catania and Rempel here back in 2002 used electrophysiological mapping to be able to map out the primary somatosensory cortex in naked mole rats and compare that to lab rats. And so what they found, this is showing a flattened cortex preparation. And so this is more rostral, and this is caudal. And in red here, you're seeing primary somatosensory cortex compared to the lab rat, along with visual and auditory cortex in the lab rat. So what they showed through their electrophysiological mapping is that primary somatosensory cortex takes up a huge amount of cortex, and also that within that primary somatosensory cortex, a very large amount of that is taken up by just the 2 contralateral incisors. So just that contralateral upper and lower incisor make up about 30% of this already huge somatosensory cortex. So that makes them a really good animal model for tactile processing, sensory processing of those incisors, and also sensory loss, because just losing one tooth is a huge sensory loss to that cortex. You're losing a lot of inputs there. What they also showed is just a brief sort of homunculus type diagram. So for humans, our hands and lips are especially well innervated. So those are represented in this homunculus. For naked mole rats, Ken Catania had an artist render this. So it might be a bit the stuff of nightmares, but it does kind of illustrate really well that these incisors are very well developed, very sensitive. And those, in addition to those tactile hairs, really make up that somatosensory array that's very important for naked mole rats. So again, we became very interested in not only this kind of dominant tactile modality for naked mole rats, but also how these weaker modalities of visual and auditory processing might be sort of still helping to bolster tactile inputs and provide additional information. So previous multisensory studies in other species have shown that these weakly effective stimuli from other sensory modalities tend to generate these neural and perceptual enhancements when they are combined together. So this is shown in a neural example over here, and then a perceptual example over here. So in this example, this is audio-visual combinations, and they are eliciting this neuronal response that you see here in this firing rate by giving a visual stimulus within its receptive field. Then that stimulus is given alone. Next, the auditory stimulus is given alone within its receptive field with this accompanying neuronal response. And then when these 2 stimuli are delivered together, auditory and visual together, you see this big enhancement of the neuronal response. And so this is greater than even the sum of the parts. So the visual and auditory presented alone. So you get this multisensory enhancement and interaction that's giving this neuronal enhancement as well. And we see this perceptually too. So this is just kind of a basic illustration where a cat is walking by a bird. It wants to be able to detect the bird. In this first situation, the bird is giving out an auditory cue, but the bird is obscured visually. In the second panel, you've got the visual cue, but no auditory. And then in the third, with both the auditory and visual cue, at this point, this reaches that level, that threshold of being able to detect and perceive the bird. So now the cat can detect it and respond to it and orient to that bird. So again, these both neuronal and perceptual enhancements when multisensory stimulus combinations are used and combined. So part of our goal in our lab is to kind of understand the role that these additional sensory modalities may play in the neurophysiology underlying dental and craniofacial sensations, and also to be able to characterize the neuroplasticity caused by the loss of normal sensory inputs from the teeth. So following tooth extraction, looking at that resulting neuroplasticity. So in a paper that we have under review currently, we are looking at and kind of talking about some of these potential multisensory stimulus combinations that may be important for tooth sensation and important in this animal model for tooth sensation and tooth loss that we're using with naked mole rats. So we created this diagram that's very similar to that flattened cortex preparation that we just saw, motor cortex being over here, somatosensory cortex, presumptive visual and auditory cortices here. And then we have different zones where the presumptive multisensory neurons would be most prevalent. So typically what's been shown in rats previously in electrophysiological experiments is that where these principal sensory and motor areas are between those regions where there's that overlap is where you would find the highest incidence of multisensory neurons. So in brown here, showing where potential sensory motor neurons would be located. In purple here, more visual tactile. In green here, audio visual. And then over here in this orange area, auditory tactile, potential high incidence of multisensory neurons there. So with this, we're interested in looking first at just kind of normal, typical, intact multisensory stimulus combinations and neuronal responses. And so in naked mole rats, we're expecting that these multisensory stimulus combinations, even with these weaker sensory modalities for naked mole rats, so the auditory and the visual being weaker sensory modalities, we believe that these will generate these stronger neuronal responses with increased firing rates compared to tactile stimuli alone, with the greatest ones being related to those very important behaviorally critical incisors. And we believe that these multisensory inputs are critical and pervasive in modulating the neural circuitry mediating tooth sensation. In addition to that, we want to look at removal of a lower incisor. And again, just keep in mind that if you remove that lower incisor, that takes away this entire region of inputs to the cortex and somatosensory cortex, so about 15% of that somatosensory cortex. So it's a big remapping. And what's been found is when one of those lower incisors are removed, it does remap the somatosensory cortex to surrounding orofacial regions. So we have done these tooth extractions in naked mole rats, which is not easy because that incisor lies really deep within that socket. And we've been able to do these as successful recovery survival surgeries. So you can see this naked mole rat down here is missing that right lower incisor. So we're really interested in looking at the neuroplasticity following the tooth extractions and using these guys as our animal model for tooth loss, and how multisensory modalities may be able to come in and help to rescue that big loss of tactile inputs. So we expect that following these tooth extractions, we might find increased numbers of multisensory neurons responsive to those surrounding orofacial regions and greater enhancements of neuronal activity related to those multisensory stimulus combinations that are really helping to kind of rescue those lost and very important tactile stimuli. So just to wrap up some of the things about naked mole rats, I just wanted to, again, kind of emphasize them as biomedical animal models and things that are kind of wonderful about them and really, really useful in considering them as an animal model. Again, their longevity, living about 40 years or more, being highly cancer resistant, considered non-aging, all of that makes them really desirable animal models. Also tolerant to those hypoxic and hypercapnic conditions. Again, for us, we're particularly interested in that tactile somatosensory system and those specializations related to the incisors and potential multisensory capabilities. They also have a really unique selective pain intolerance to acids that's been shown. And then also just wanted to briefly touch on some of those limitations and kind of special considerations in considering this as an animal model. So they are functionally blind, a bit with poor with hearing and sound localization. They do have some issues with thermoregulation that need to be considered when designing experiments for them, behavioral experiments and such. And also their eusocial colony structure can hinder them being isolated for long periods of time, and food and water restrictions when trying to kind of motivate them to learn a task. They do get all of their water through their fruits and vegetables, so can't really remove water from them without removing food and so on. They also tend to attack any non-colony members immediately. They're pretty vicious with that, and with all that head musculature related to their bite force, they can do some damage with that. So again, a wonderful animal model, but some things to consider as well related to their specializations and their adaptations. So with that, I just want to thank all of my past and current lab members who've contributed to all of these studies, and especially the American Physiological Society and Fauna Bio for hosting this and inviting me to speak with you today. I really appreciate the opportunity. And in addition to that, I want to thank the NIDCR for help with funding this research on the multisensory integration related to tooth loss. Thank you.
Thank you so much, Diana, for the great presentation. Now at this time, I'd like to call Dr. Ben Sajak and Dr. Amaral Silva back onto the screen for our Q&A session. And just a gentle reminder to our audience members that if you have any questions for our speakers, please feel free to submit them using the Q&A function at the bottom of your screen.
Okay, our first question is for Lara. Does the age of the chick influence the firing pattern of neurons to hypoxia?
So possibly, we are still at the very beginning of this kind of research, but when I was looking for preliminary data, I did have a range of ages, so from 5 days old to 6 weeks, and we are still troubleshooting this, but possibly, but not very clear.
Not seeing additional questions yet. I have one for Diana. Have you seen any colony consequence to the tooth extraction? You said there's like, they're infighting for non-colony members. Has there been any like social isolation or anything noticeable if the tooth is extracted?
Yeah, we haven't seen that. That's a great question. One thing that we were interested in tangentially was to kind of assess the entire colony, kind of establish what their entire social hierarchy is, and then do tooth extractions and see if that made a shift. So and so has lost a tooth. Well, he gets knocked down.
Demoted. Yeah.
Knocked down the social hierarchy a bit, and he's got to do some of the lesser work or something like that. So we are a bit interested in that, but mostly one thing is that when we do sort of long surgeries or things like that, we do worry about them coming back sort of smelling a little bit different and then possibly being attacked. So we try to be very, very careful about that. But otherwise, we're kind of assessing some of that behaviorally as well.
Cool. I got another question that's come in for Diana. Are the naked mole rats able to regrow the lost tooth? Maybe the one that's extracted?
Yeah, that's a great question. They are not when we completely extract the tooth. So in that one image I had where it was all the way back to the root, if we sort of pull that entire thing out successfully, then they are not. So otherwise they would be continuously erupting teeth that constantly grow and need to be worn down. But with our extractions in the adult animals, as long as they are complete and we get the entire thing out, including the root, then those do not regrow.
Do you have to trim those regularly? Or if do they just keep growing? Like I have to trim my rabbits,
Yeah. Yeah, for the most part, they kind of self-regulate on
take
that.
care of it.
They chew on things all over the colony. We do have some now and then that get very overgrown incisors, and that's actually what I had to do on Friday on our snow day, kind of come in quickly anesthetize an animal and trim those upper teeth because it wasn't eating as well. So sometimes they need a little bit of help, but in the wild and in our lab mostly, they seem to find things to chew on. That's part of their enrichment as well. They really, really enjoy chewing on anything they can get their teeth on.
Very interesting. Well, love the models. Not seeing any other questions come in. I think I'll pass it back to Jacob, and we're a little bit over, so we'll wrap this thing up, I think. Oh, we have one more question that came in, Jacob, if you don't mind. What is the level of sensation in case of damaged incisors? The level of sensation.
I'm not sure I understand the question.
So if they're damaged, do they lose sensitivity? Maybe that's another way of putting it.
Okay, yeah, I would say we don't quite know the answer to that in terms of their ability to sort of investigate and exactly how sensitive each incisor is and what they're doing with each of those individually. I think that's kind of the question we're trying to get at with the innervation for one thing, but in terms of remapping the cortex, of course, those other, just like with say an amputated limb for us or something like that, surrounding areas sort of take over cortically with that neuroplasticity. So within the central nervous system, those areas can kind of come over and help take over. But yeah, in terms of the explicit sensitivity of individual incisors and what they're capable of, that's still an area of great interest.
Okay, and I think we'll take one last one here. I think this is again, in response to the naked mole rat, what is the translational potential for your research?
Yeah, thanks for that question. So for some of this, we're just really interested in sort of the basic science aspect of it. So just looking at these animals and their unusual adaptations. But in terms of their overall translational potential, obviously they have a number of different things related to aging and cancer studies, and also those tolerances of toxic conditions and so on. They also live that long without neurodegeneration. So in looking at some of these aspects and with the tooth loss, as in particular, so the NIDCR is particularly interested in that, and having naked mole rats as that animal model. In humans, the tooth representation is very small. It makes it hard to study. And also you can't just take a human's tooth out whenever you want. So the timing of it in terms of the age, the interventions that you do, we're really interested in some multisensory stimulus strategies that can come and help rescue some of the deficits that are observed in terms of some things that happen when humans lose their teeth. So they especially have issues with social interactions. They become self-conscious. They have issues with chewing and speech. So some multisensory stimulus combinations may be useful, and some therapeutic approaches for that as well.
Okay. Thanks for all those questions and the great responses. Now I'll pass it over to Jacob to wrap up.
Thanks, Ben, and thank you both for those wonderful presentations. We just want to thank Fauna Bio again for supporting our mission to provide, great scientific education. I also want to thank our audience for joining us today and spending your late morning and early afternoon with us. Before you sign off, we hope that you will take a few minutes just to complete a short survey about today's session and any ideas that you may have for future comparative physiology research programs that you'd like us to cover, different areas that you'd like to see us cover in our next session, just please let us know. All right, well, with that, I want to thank everyone again, and we'll see you next time. Take care.
Thank you.